Good morning. This is Hope Reactor. Now, let's begin today's news.
The cancer letters in our blood just became 25 times easier to find. Cancer cells may be able to recognize cells of the same type. Now researchers are using that unusual behavior to make tiny, cancer-related signals in the blood easier to capture. The key is the exosome, a microscopic package released by cells. By modifying the surface of these packages with rare earth ions, researchers increased their ability to bind to the same type of cell by more than 25 times. If this technology continues to develop, it could support earlier cancer detection, targeted drug delivery, regenerative medicine, and even research into aging. What we don't know can become hope. This is Hope Reactor. Our bodies are made of tens of trillions of cells. Skin cells, muscle cells, nerve cells, and many others all perform different jobs. But they do not work in isolation. Cells are constantly exchanging information. One way they communicate is through exosomes. Exosomes are extremely small membrane-bound packages released by cells. They are usually only tens to hundreds of nanometers wide. Inside, they can carry proteins, lipids, and molecules related to genetic information. A simple way to imagine an exosome is as a tiny letter, sent from one cell to another. Healthy cells send their own letters. Inflamed cells send different letters. Cancer cells also release letters that reflect what is happening inside them. That is why researchers are interested in studying exosomes in the blood. If we can identify where these packages came from and understand what they contain, they may reveal important changes occurring inside the body. But there is a major problem. Blood contains enormous numbers of particles and exosomes, released by many different types of cells. Finding the exosomes released by one specific cancer is like searching an ocean for a single message in a bottle. And the earlier the cancer is, the smaller the signal may be. We want to detect cancer as early as possible, but early signals are often the hardest to find. To overcome this challenge, researchers focused on a natural feature of exosomes. Exosomes can show a preference for binding to cells, similar to the cells that release them. In other words, they may behave as though they remember where they came from. An exosome released by one type of cell may be more likely to attach to or enter that same type of cell. The researchers tried to strengthen this natural targeting ability. They modified the surface of exosomes using rare earth ions. Rare earth elements are used in many technologies, including electronics, magnets, and optical materials. The important point is that the researchers did not completely rebuild the contents of the exosome. Instead, they changed the outer surface, the part that first comes into contact with a cell. It is a little like keeping the message inside a letter unchanged, while redesigning the envelope so the intended recipient can recognize it more easily. After this surface modification, the exosomes showed more than a 25-fold increase in binding to cells of the same type. That number matters because stronger binding could make weak biological signals easier to collect and measure. If only a tiny number of cancer-related exosomes are circulating in the blood, they may normally disappear into the background noise. But if their natural attraction can be strengthened, researchers may be able to concentrate those signals and detect them more clearly. In the future, this approach could support liquid biopsy, a method that looks for signs of disease in blood or other body fluids. Unlike a traditional tissue biopsy, a liquid biopsy may reduce the need to remove tissue directly from the body, a more sensitive way to capture cancer-related exosomes could eventually help doctors detect changes earlier and monitor disease with less burden on patients. However, this does not mean that a new hospital-ready cancer test has already been completed. The result is still part of ongoing research. Real blood is extremely complex. It contains cells, proteins, fats, and many kinds of microscopic particles. Researchers still need to confirm safety, stability, accuracy, and whether the method can distinguish the intended target from other cells. They must also determine whether the technology can be produced consistently and used reliably in real clinical samples. So this is not a finished medical test. It is a promising foundation for future diagnostic and treatment technologies, and its potential may extend far beyond cancer detection. The first possibility is finding disease-related signals. If rare exosomes can be collected more efficiently, doctors may one day detect biological changes that are currently too faint to see. The second possibility is targeted drug delivery. Exosomes can be explored as natural delivery capsules. If medicine is placed inside an exosome that strongly binds to a particular cell type, it may help carry treatment to the place where it is needed. That could improve effectiveness while reducing unnecessary exposure to healthy cells. The third possibility is regenerative medicine and aging research. If helpful molecules can be guided toward damaged cells or aging tissues, exosomes may become part of new strategies to support repair and recovery. What makes this research especially interesting is its approach. Instead of forcing cells to behave in a completely unnatural way, it strengthens a targeting ability that already exists. It works with the body's own communication system. At this moment, countless microscopic messages are moving through your bloodstream. Until now, many of those messages have been too small and too mixed together for us to read clearly. But if scientists can guide them, collect them, and amplify their signals, the same tiny packages could help us find disease earlier and deliver treatment more precisely. A behavior used by cancer cells may become a clue for detecting cancer. A microscopic cellular habit may become a powerful tool for future medicine. Would you take a blood test designed to find these tiny warning signals before symptoms appear? Let us know in the comments. Hope Reactor explores new science and technology and turns complex discoveries into clear stories about the future. Subscribe for more ideas that may change how we see medicine, technology, and possibility. What we don't know can become hope. This is Hope Reactor.